Before I start presenting about my topic, I have a question for all of you. This is three scientists who awarded the 2015 Nobel Prize in Chemistry, and the question is, do you know the Nobel Prize 2015 in Chemistry was awarded for what? You can comment on the box or open micro to answer. Any answer? Okay, thank you. That's the right answer. The answer is mechanism of preparing DNA, and this mechanism plays role in study how cancer treatment. But we are just students, so before understand the complexible mechanism, we must find out some basic knowledge about DNA or nucleic acid. And that's the topic I am going to talk today. I will present about DNA replication, transcriptions, and medical application. The first one is DNA replication. For simple, DNA replication is the process producing two identical replicates from one original DNA molecule. This process occurs in all living organisms and is basic for biological inheritance. And the question is, when will DNA replication occur? You can see interface here. This is the longest period of the cell cycle, and DNA replication occurs in the phase of interphase. Next is the enzyme. You may find the name of the enzyme are very familiar. We have none is in the high schools. This enzyme will catalyze various steps in this process. For example, DNA ligase will unwind and require DNA change to prevent the DNA from becoming subcoiled. Besides the enzyme, we also have some components are needed, such as SSB proteins, binds and prevents DNA double helix recoil after DNA replicate in Y cells. Remember this enzyme, and I will talk more particularly about their function when going into the process. The rules there are two main principles to keep in mind are replications is a semi-conservative process. This means a new DNA consists of one change from mother and the other is complementary from environment. The second is specificity of base pairing. This means adenines will connect to thymine by two hydrogen bonds, and guanines will connect to cytosine by three hydrogen bonds. Now is the most important thing. DNA replication has three stages: initiation, elongation, and termination. And I will present through this video. The first step is separate the two chains. Enzyme unzip a helicase. It makes a replication fork. After separate, each chain will be a template for creating a new chain of DNA, and DNA ramase will generate RNA rimers. Rimers are short RNA molecules that mark a starting point of DNA replication. After that, DNA polymerase trees bind to the rimer and make new chains follow complementary principle. And this enzyme has only one direction, 5' to 3'. This is very important. It creates one reason that we have two kinds of chains. Leading chains is made continuously and lagging chains is run in opposite direction. Therefore, it's only made from series of small fragments called Okazaki. Next, in elongations, the rimers are formed by DNA ramase and DNA polymerase III continue each other, and this process will repeat again and again in other positions. Once new chains emerge, exonuclease will remove or write RNA rimers from both chains of DNA, and the DNA polymerase I fill this gap. Finally, DNA ligase will connect fragments to form a continuous double chain. That's all about DNA replication. The second part is transcription. Transcription is the synthesis of RNA molecule by using a DNA molecule as a template. It transfers the genetic information from DNA to RNA. And when this process occurs, it's different. In eukaryotic cell, it occurs in nucleoid regions, and in eukaryotic cell, it occurs in nucleus. Transcription also has three stages. In initiation stage, RNA polymerase bind to special regions in DNA called promoters. In humans, promoter is GA box. This unwinds the DNA double helix and provides single chain template in 3 fragments to 5 fragments need for transcription. Transcription starts from here in the initiator site. In elongation state, RNA polymerase move along the template DNA chains in 3 fragments to 5 fragments by an mRNA out of the complementary space pair. You can see messenger RNA is have direction 5 fragments to 3 fragments. And finally, the termination. Once the DNA polymerase by two sequences called terminators, the transcription stops. The RNA molecule and RNA polymerase are released. Which region in genes is transcribed already will rewrite immediately. Now there are some differences in progressing mRNA we have to note. In eukaryote, the RNA transcripts are ready to be used directly as a template for translation. And in eukaryote, the RNA transcripts are called re-mRNA and must go through extra resin called RNA lysing before it can direct translation. For particular process, look at this video. In eukaryote, the mRNA has many intron and extron. The mRNA will be added of 5' caps at the beginning and the 3' poly tail at the end. Then lysosome will remove intron and extron are stuck back together to make the major mRNA that leave the nucleus, enter the cytoplasm and translation begin. You can see the cap and the tail are protecting mRNA when it goes through nucleus spore. Now we move to transcription factors. Transcription factors are proteins that regulate the transcription of DNA into RNA. They control the beginning of transcription. RNA polymerase can attach to the promoters only with the help of them. Some transcription factors called activators that activate transcription. They may help the basal and RNA polymerase polymerase bind to the promoter. Other transcription transcription, they are regressor. The regression can work in a variety of ways. So this important protein can help determine which genes are active and in each cell of your body. In addition, some group of transcription factors, binding sites called enhancer and silencer can turn a gene on or off in specific spots of the body. Next is RNA polymerase. Is the enzyme that is responsible for copying DNA sequence into RNA sequence. Like DNA polymerase, RNA polymerase catalyze the formation of ester bonds between nucleotides that pair with the complementary nucleotides on DNA template. But unlike DNA polymerase, RNA polymerase initiate the synthesis of new chains in the absence of primer. And the final problem I want to talk is type of RNA polymerase. Difference between RNA polymerase of eukaryote and eukaryote. It is summarized in this table. And you can see in the other table, RNA polymerase also has three types, like DNA polymerase. Next part is medical applications and book will be sent to you.
Hello everyone. So following the presentation, I will talk about how we use this knowledge to apply in clinical. First, we move to applying in diagnosis. There are two things that I think really important. That is PCR and recombinant DNA. First, PCR. PCR is stands for polymerase chain reaction. It can detect rapidly and accurately the residence of bacteria such as chlamydia, mycoplasma, mycobacteria, herpesvirus, and enterovirus directly from clinical specimen. Its components are nucleotides, rammer, Taq DNA polymerase, reverse transcriptase, and thermocycle procedure. PCR involves four steps. First, we need to extract nucleic acid from the microorganism. Next, we will naturation at 90 to 95 Celsius degree. We will separate the extract double strand DNA into single strand. The third step, that is annealing, we cool into 55 Celsius degree. We cool into a low rimer, as you can see the black one here, to bind to the single strand that we separate. And the last step, that is extension. The enzyme Taq polymerase and nucleotides are added to create new DNA fragments complementary to the target DNA. And after that, we will repeat a lot of cycles, and the result is an accumulation of specific target sequence. As you can see here, a lot of target sequence. That's PCR. Next, we move to recombinant DNA. But first, before we go into detail, I need you know two fundamental difference between PCR and recombinant DNA. Recombinant DNA is the replication of the DNA within a living cell, and it's cutting and pasting DNA sequence. In contrast, PCR is the replication DNA in the test tube, which is free of living cells, and it is amplified by copying an existing sequence. Recombinant DNA has 7 steps. First, we need to choose the host organism. As you can see here, the host cell. Next, we will repairing a vector, repairing a DNA. Next, we will cutting vector and DNA by restriction enzyme. Then we will combine it together and create the recombinant DNA. That is the step 4. Step 5, we will introduce it into the host cell. Next, we will selection of organisms that containing the recombinant DNA. And the last study, we will screening for lungs with designed DNA insert and biological properties. So how we use recombinant DNA in our career in the future? Recombinant DNA is used to reduce human insulin, human GH, and blood protein factor 8. We also can use this to diagnose genetic disease, then diagnose microorganism infection like HIV, or maybe diagnose in tumor pathology like leukemia, lymphomas. Yeah, and that's all about diagnosis. We move into treatment. The first target in treatment is hepatitis B virus. This is the structure of the virus that we have learned in microorganism. This virus will affect the liver cells, as you can see here, and it will through a lot of steps to generate their to create their new generation and also damage the liver cell. So if we use drugs that terminate those of this virus step, this virus will not harmful with our body. So that is the work of nucleotide analogues. How it works? It will fool the hepatitis B virus into thinking that they are normal building blocks for DNA. Next, we will, we will use this to treat cancer. How they work? This agent behave as anti-metabolite, compete with physiological nucleosides, and interact with a large number of intracellular targets to induce cytotoxicity. We have some example in here. Pyrazolopyrimidine or allopurinol for short is used for treat high uric acid in blood or gout because it inhibits synthesis purine and xanthine oxidase action. Nucleotides contain arabinols, which is used for cancer treatment and viral infection. 5-iodo-2 deoxyuridine is used for keratitis by herpes. And the last one, that is 5-fluorouracil, is used for cancer treatment. Next, we go to nucleotide excision repair. There are 3 excision repair pathways exist to repair single-strand DNA damage. That is nucleotide excision repair, base excision repair, and DNA mismatch repair. So DNA damage occurs constantly because of chemical, because of radiation, and also because of UV light, ultraviolet. So with this technique, we can recognize the damage by UV lit, and then they will remove the short single-strand DNA segment that contain a lesson. So as you can see here, this is the lesson which is created by UV light, and after a lot of sequence, it will remove this lesson, and the undamaged single-strand DNA. As you can see here, we use DNA polymerase to synthesize the new shortened complementary DNA. The last one is gout. So what is gout? Gout is a systemic disease that results from the deposition of monosodium urate crystal in tissue. So why their deposition? There are two causes. Maybe that they are over radiation, or maybe they are under expression. With under expression share a major percentage, about 90%. Over radiation, this come from dietary bearing from your food like meat, seafood, or beer. Endogenous bearing synthesis and malignancy, tumor lysis syndrome, which is mentioned before. Burying civet, that's HGBRT deficiency. This is an enzyme in the burying civet, and when it deficiency, it causes hyperuricemia. And the last one is burying retina. You can see in glycogen storage disease. In under expression may come from you use diuretics, maybe your kidney uh, has some problem in renal failure, or maybe you consume a lot of alcohol, you have a genetic defect. All of this and with this risk factor like male age and obesity will create hyperuricemia, and it will increase the renal and gastrointestinal tract excretion. So how we treat it? We will use allopurinol and oxypurinol. Both inhibit xanthine oxidase, an enzyme in the burin catabolism pathway that converts hypoxanthine to xanthine and to uric acid. Adverse effects: it will increase incidence of acute gouty flares, maculopapular pruritic cracks, maybe gastrointestinal adverse effect including nausea and diarrhea. So that's all. This is the member list of our team, and this is the reference that we use for this PowerPoint. Thank you for your listening and hope you enjoy it.
Thank you.
Thank you.
Alo, mọi người có nghe rõ không vậy?
Có.
Okay. Okay, hello everyone. Here we come to the presentations of group 4 about specific metabolism of nucleic acids. And our instructor is Hà Thị Thảo Mai. These are our members. Today we have 6 parts total. And the first one is uric acid metabolic disorder. So we should find out the definitions of uric acid. So uric acid is the final product in the process of degradation of purine in plant, red meat, seafood, and alcoholic beverage. The enzyme deficiency is hypoxanthine phosphoride oxide transferase. We can call XBRT for short. It's one of the central enzymes that make nucleotide from base purine. And look at this image here. You can see that XBRT is very important to synthesize nucleotides. If XBRT decreases, it will become to uric acid. And the consumption of high purine food will make the kidney cannot remove uric acid. And when uric crystals are deposited too much in the kidneys, forming kidney stone, glomerular fibrosis, interstitial nephritis, especially this overproduction of uric acid we can call hyperuricemia, leading to gout. And gold patients have hyperuricemia in the synovial fluid, leads to the formations of urate crystal precipitate. And when the cartilage is damaged, it will attack to the bone below and leading to trophy cyst formations. And the final is will destroy the bone. And the second part today is adenosine deaminase is standard for ADA. ADA catalyzes the amination of adenosine to inosine. Here, about specific features, ADA is produced by cells throughout the body and is associated with the activations of lymphocytes. For example, Mycobacterium tuberculosis cause increase the amount of ADA. An ADA test measures the amount of adenosine, aden, the aminase present in pleural fluid, so the doctor can diagnose a tuberculosis infection of the pleural. We must be patient that ADA test is not a diagnostic test and does not provide for the cultures of diagnosing tuberculosis because it may be positive even when the number of bacteria are very low. The test is recommended in some cases, like migrated from blood spot, pregnant people, be in close contact with someone who is suffering from tuberculosis or have a very low immune system, children who have positive in the tuberculosis screening test, who in the hospital are social worker also. Okay, let's move to DNA nanotechnologies. DNA nanotechnologies is the design and manufacture of artificial nucleic acid structures for technological use. Nucleic acids are used as non-biological engineering materials for nanotechnologies rather than the carrier of genes, and forming DNA to two-dimensional and three-dimensional structures for medical purpose throughout four structures below. And one of these DNA structures, DNA's origami, is the nanoscale, folding up DNA to create two-dimensional and three-dimensional shapes. The long single-stranded DNA is folded into the desired shape by multiple short single-stranded DNA. So we can realize that DNA is used as scaffolding for holding other molecules in place or to create structures. And about its applications, the first one is pathology resolution to improve controls of neuron prosthetic and prosthetic limbs, or smooth the irregular neuroelectrical activities, or amplify neuronal signaling. The second application is making smart drugs for target drug deliveries, as well as for diagnostic applications. This image is about nano capsule, and this one is about treatment of respiratories, and this one is about treatments of pulmonary cystic fibrosis. And the final applications is environmental support in neuronal repair and activate neuronal array. Okay, let's talk about genetic engineering. Genetic engineering is the direct manipulation of an organism genes using biotechnology to change the genetic structure to make biological products or better organisms by using recombinant DNA methods or by artificially synthesizing the DNA to change the genetic material. And genetic engineering have numerous fields in fixing severe genetic disorder in humans, just vaccines and other products from organisms for crop and in crop for nutritional values and tolerance to environmental stress. Okay, we go to recombinant DNA. Recombinant DNA technologies is the churning of DNA molecule from two different pieces here. And here are the products. The recombined DNA molecule is inserted into a host organism, and in this image, the host organism is bacterial cells to produce more and new genetic combinations. And about applications is to identify, map, and sequence genes and to determine their functions, or analyzing gene expressions, or examining protein synthesis in cells and organisms. And other applications, recombinant proteins is to modify gene sequence and useful products in human insulin, growth hormone, or blood clotting factor 8, hepatitis B vaccine, etc. Okay, we go to DNA fingerprinting. And as you know before, this is what makes DNA fingerprinting as a useful tool. It allows us to identify an individual from biological samples. It's called fingerprinting. And the applications is physically connect a piece of evidence to a person or rule out someone as a suspect, show who your relative, or identify a dead body that is too old or damaged, match tissues of organ donor with transplanted people. And as you can see, this have total of 6 steps. Step 1 is extract DNA nuclei from the cells. Step 2, they use PCR for amplifying the DNA. Step 3 is to cut DNA strand into fragments in specific positions by restrictions enzyme. Step 4 is sorting the DNA segment by Chell's electrophoresis. Because of the negative charge molecules, the shorter segment the farther is moved away or original locations. And step 5 is loading the DNA gel onto a suitable membrane. Single-stranded DNA fragments are transferred on the membrane. This hybridized with a suitable DNA growth. The DNA to be investigated like DNA found at the prime site. And before these probes are used, they are tagged with a radioactive substance or fluorescence dye. And the final step, we can see the result by autoradiograph. And this is a technique using X-ray film to visualize molecules or fragments of molecules that have been radioactively labeled. And the final part today is SNRNBS and human disease. SNRNBS is a class of small RNA molecules found in nucleus of eukaryotic cells, approximately 150 nucleotides, and the most common human snRNA component is included from U1 to U6 except for U3 by awesome RNA. And the goal is to strengthen the mRNA here, here, into a mature and fully functions mRNA by cutting the intron and splicing the exon together after that. Okay, about human disease. The consequence of spinal muscular atrophy is muscle weakness, poor muscle tone, and the causes is the mutations in the survival motor neuron 1. This keratosis congenita is the mutations in the assemblage SNRNBS. This disease will express in skin, nails, and mucous membranes. The last one is Prader-Willi syndrome. Prader-Willi syndrome will make the patient feel extreme hunger, confusion, and behavioral related to the making of our presentations. Thank for your listening. And do you have any questions?
Không ạ.
Cool.
Okay, goodbye. Thank you very
Rồi.
much.
Thank you.
Thank you.